Good morning. I guess I started the idea core at a perfect time, just when we all went into a pandemic. And it was a challenge, but we managed to accomplish a few things that I want to share with you all today. The idea core is I think the easiest way to describe what it is I do is just to share a few things that I've done in the past that led me to start the core. So we offer a handful of services, modifying an existing apparatus or data analysis pipeline for mostly neuroscience labs, replicating a previously published apparatus that maybe the lab that needs to use a device either doesn't have the skill set or the time to reproduce the apparatus. I also help design, assemble, and deliver a novel apparatus. Let's say a lab has an idea for an experiment that they want to do, but it's never been done, and there's nothing on Amazon that one can just order and use. I also am available for consultation or assistance analyzing data. Oftentimes a lab will come to me, they have a feeling that their data could be analyzed in MATLAB, but they've never used MATLAB before, for example. So some of my software skills have been useful. So let me just introduce some, let's see, a device that I developed in my postdoc. I worked at a neuroscience institution in a fly lab, and I developed a device that was used to record flies escaping from a simulated predator. We called it FlyPez because it releases flies one at a time, much like the candy. And the flies, this is a 3D model of the device. A vial of flies is placed in a position that allowed the flies to walk upward onto a viewing platform, and a laser shines through the tunnel to sort of convert the image of the flies into a one-dimensional image. A gate would close to block subsequent flies, and the fly on top would be filmed in high-speed video. At the end of it, they would be discarded if they didn't jump away from the simulated predator. Lights were also used for optogenetic stimulation. Guidon introduced that concept earlier. And then small mirrors would send two viewing angles to a single camera. Behavior was induced either using the optogenetic lights or a 3D panoramic visual stimulus that surrounded the animal. So that's the dome that was a 3D or a panoramic screen, and a projector above the device would show a visual stimulus for the animal. There were 4 of these devices running all the time in the lab. They're still using them. And oh yeah, slide please. And the whole purpose was to make the experiment fast and easy. So this is Pat. Pat was a technician in the lab. High-powered LEDs were used for the visual stimulus, and we had a graphic interface that was used to start and stop the experiments. A water moat kept the flies from just walking away. and a barcode was used to keep track of experimental parameters and details about the genetics of the flies that were being experimented on. So this is a real-time view of the fly coming out and everything else that happens. We'll slow it down so that you can see each part of it. So you can see the 2D representation of the 1D of the fly coming up through the tunnel. After it passes a threshold, the gate closes and the animal emerges onto the viewing platform. And this is a live feed that one can see on the computer. In some cases, we would try to scare the animal to make it jump away or use optogenetics. And then the high-speed version of the video is downloaded, and if it doesn't jump, it gets swept away. So this goes on for 20 minutes, during which we would get about 20 videos per run and 20 runs per day per fly pez and 4 fly pezes. So there's a lot of data to manage. So I also generated a data management pipeline where the users would curate each video, make sure that the data looked good, it was usable. And that's what I did for my postdoc. So recently in neuroscience, a kind of, so systems neuroscience is an emerging popular field. And this type of neuroscience looks at populations of neurons and often requires the scientists to design creative experiments that involve behavior. As the complexity of the neuroscience question increases, oftentimes the complexity of the experiment increases. One needs to observe different types of behaviors in very controlled settings. And it seems that frequently the ideal experiment requires some kind of technology that simply does not exist. And so I've had some experience generating these types of technologies, and each project has been largely different and unique, but you know, to generate the device, it requires a skill set that I have, and that's what the core provides. So this is an example of a head-fixed panorama. A mouse would sit in the viewing space of this screen, and then a moving grating was presented to stimulate the eye of the animal. A couple of cameras record the movements of the eye, and at the same time, the experimenter is recording from the brain. In this case, Steven Santoro approached me and said, you know, there's a published experiment that requires equipment that we don't have and don't know how to assemble. And we have a couple of old pieces of equipment that were used for a similar experiment. You know, can you help us modify these things for the purposes of our lab? And so I generated a foot shock inducing box, and there's a camera that records the behavior of the animal, and a couple of scintillation vials that are delivering an odor at the time of the foot shock, and they're looking at the way certain types of stem cells express in association with the odor and the foot shock. In another case, a lab approached me and said, we want to record from the sciatic nerve of rats, but we've never done the experiment or any electrophysiology, and we don't have the equipment. So in this case, I helped them just assemble the appropriate pieces of equipment, and we've gotten to the point where we have data that was used recently for a grant for looking at nerve regeneration in the sciatic nerve of rats. So what I bring to the table is a skill set that includes fabricating and designing novel pieces of hardware. I'm fluent in MATLAB and Python, and I have experience in scientific research, which I think helps me bridge a gap that sometimes exists. I function as a bit of a liaison between science and the engineering world. You can email me at ryan.williamson@curanschutz.edu, and I also have a website on the, it's a child website under the Neurotechnology Center website on campus.